Good morning, everybody. All right, so today is really just a little bit of finishing up the end of homologous recombination. Let me just make sure I know where I'm at for that. And then it's really going to be a case of, fire away any questions and we'll just, see what things we need to talk about. Okay, so that's where we're at. Okay, so here we're at. Remind me again, I couldn't see from the video. Have we gone through homologous recombination in full yet? I can't quite remember from the last class. Can't remember what I was doing, 5 minutes ago, to be honest, other than making a cup of coffee. Did we finish going through homologous recombination? Because on the video, this was where we ended up, right here. Did we go any further than that?
No, we didn't.
Oh, okay, great. Thank you. Is that Jelani?
Sophia.
Oh, Sophia. I'm sorry. You sound
It's
very
okay.
similar. Okay, so before I get to that, I do remember saying I'd look up DNA polymerase alpha. Where are we? Which is this one. So DNA polymerase alpha is both a primase and a DNA polymerase, right? And so the difference, well, one of the differences between prokaryotes and eukaryotes in DNA replication is in prokaryotes, you have a primase, which is an RNA polymerase. It's an enzyme on its own, which synthesizes RNA primer, and then you have one polymerase. Come on, where are we at? Here we go. DNA polymerase 3, which does all of the DNA synthesis, right? Apart from removing the primers and replacing them with DNA, right? So in eukaryotes, it's different, right? Basically, in eukaryotes, we have, let's make sure I get to the right, one DNA polymerase that starts the process in both making the primer and synthesizing a small stretch of DNA, not very long. I actually don't know exactly how long, but it's pretty short. And then we have one DNA polymerase for the leading strand, which is epsilon, and one for the lagging strand, which is delta, right? And those do the whole rest of the DNA synthesis, right? And so one of the questions that I needed to ask for my own, benefit was, how does DNA polymerase alpha do that, right? Because I always thought DNA polymerases can't synthesize RNA and they need the RNA primer to do their job. Well, the answer is that DNA polymerase alpha is actually made up of a bunch of different proteins. It's made up of 4 proteins. These are called subunits, right? So this is different from a hollow enzyme, right? So if you remember, we talked about, I'll just kind of write that up again. Oh, dang it, if I can actually spell it right. And so really a hollow enzyme is a complex of enzymes, right, that come together to perform a function. Whereas what we have with DNA alpha is this is actually a multi-subunit protein, right? And what that is this kind of seems a little bit like semantics, but it is an important difference, is one protein made up of two or more proteins. And those are called the subunits. Right, so each of those subunits, each of these smaller proteins here is encoded by a separate gene, right? It's not one gene for the whole of DNA polymerase alpha, it's 4 in total. But these only, when these are synthesized, they kind of just fit together, right? Automatically assemble in the right shape, right? It's not that they kind of come apart and then go back together again and come apart and go back together again like holoenzymes do, depending on the need. DNA polymerase alpha is one protein, right? It just happens to be made up of different parts that are not encoded by the same gene, right? So that's kind of the difference between holoenzymes and multi-subunit proteins, right? And a lot of proteins are made up of more than one subunit. Have a think. Hemoglobin, for example, is made up of 4 different proteins. Histone, nucleosomes, right? The protein complex made of 8 different proteins, right? So you get a lot of multi-subunit proteins in biology. Anyway, that aside, the reason why DNA polymerase alpha can both synthesize prime RNA and DNA is that it's done by different parts of the protein, right? So these 3 proteins here together, I don't know why they're called P something, some kind of naming convention, right? I think it's probably based on their size, would be my guess. These 3 together synthesize RNA, right? Which you can see kind of up here, right? So the very beginning, this part of the protein makes RNA, and then once that RNA primer has been synthesized, these 3 bits don't do anything more, and essentially the synthesis is taken over by this subunit, which then uses DNA nucleotides, pardon me, to synthesize a short stretch of DNA. Then this DNA polymerase alpha, the whole thing falls off, and then either delta for the lagging strand or epsilon for the leading strand come in, and you know, off to the races we go, kind of thing, right? So that's how it does that, which is kind of neat. That is something that I didn't know a few days ago, which is kind of cool. One of the reasons why I love doing my job. So I get to learn lots of stuff. Okay, so a little bit of a tangent, but a fun one nonetheless. Okay, so talked about recombination essentially occurring in either meiosis 1, specifically prophase 1, right? When you have your fully replicated homologous chromosomes lining up. But also it can occur in dividing cells when double-stranded breaks occur, right? And double-stranded breaks are pretty literal, right? It's when both backbones of the DNA are broken, typically in the same place. Now, this is if it doesn't occur intentionally, right? If this is accidental, typically due to ionizing radiation, right? So gamma radiation, maybe X-ray radiation. I'm not sure how ionizing X-rays are. I mean, they're not great. If one of those energy particles hits DNA, it can contain enough energy to break both bonds on both sides of the double helix. And if that happens, that is terrible, right? And for a bunch of reasons. One, it will break a chromosome in 2. So if that isn't fixed, when you go through another round of cell division, you'll end up with 24 chromosomes instead of 23, which is also not good. Why else? This break might be in the middle of a gene, which would render that gene non-functional, right? So that would be a complete loss of function. And also, if this is repaired kind of ad hoc, right? So essentially it's like if you have a long piece of string, right, and you cut it in a bunch of places, if that is one uniform length of piece of string, how do you know which end of which bit goes with which, right? You don't. And so the process of repairing these breaks often introduces other problems as well, right? So double-stranded breaks are pretty calamitous, to be honest, for your cells. And that's in a large part, and especially dividing cells, right? And so that's in a large part why if you are exposed to ionizing radiation, then the cells which show, or the tissues which show problems are ones which are sites of active cell division, such as your guts, your skin, your hair, things like that. The same for chemotherapy, actually, right? And actually, this is the one reason why it's kind of scary, the thought of, say, traveling to Mars, right? So it's a big scale, right? We're not on a little scale anymore. Because people who travel to Mars would be exposed to an enormous amount of ionizing radiation from the sun, right? We're protected by our ozone layer and our atmosphere, and also the magnetosphere around our planet, right? Which is what causes the northern lights, for example, right? When there's a solar flare and stuff like that. And so this is a kind of problem that will occur most likely on people who are spending a long time in space. Anyway, that's a big tangent. I'm not going to go down that road too much. So you can fix, if your cell is in the process of dividing and you have an identical copy to match this broken strand too, you can actually fix this, right? And you can fix it very, very well, right? By homologous recombination. But typically when we talk about homologous recombination, we talk about it in the context of meiosis, right? Because that's when it always occurs. Anyway, so I can't believe I actually talked about traveling to Mars in a genetics class. Isn't that neat? It all comes down to genetics eventually. Anyway, so the very first step before this is to line these chromatids up perfectly, right? This is utterly essential because if they don't, you'll end up, if you do it like that, right? You'll end up this one getting a longer piece than it should do, and this one losing a piece that it should already have, right? And so these two have to be lined up perfectly. That's done by something called the synaptonemal complex, which I don't actually fully understand how that works, to be honest. Right, but however it does it base by base perfectly. So that, let's get that back down. So that when this break occurs, these two strands are lined up, right? That's super, super important. And so the very first step is to basically chew back some of this DNA so that we have single-stranded DNA on both sides of the break, right? We're going to need that because essentially one of those strands, now that it's single-stranded, can actually base pair with the other one, right? With its matching partner on the other chromosome, right? This is called strand invasion, right? So once you've kind of chewed this back, now this strand is going to be complementary to the other strand on the other chromosome. And now it can actually base pair. And again, because these things are lined up perfectly, the sequence of bases in this strand is going to be exactly perfectly complementary to the one in this strand, right? Or almost exactly perfectly complementary. There may be like the odd one or two bases because these are between homologous chromosomes, not between identical ones, right? And so that free 3' end invades that strand. And so now you have this weird complex right, which basically consists of 3 strands of DNA. You have 2 which are base paired together, like a double helix, essentially, right? And you have a third one which is essentially kind of flapping in the breeze, right? Because it is no longer base paired to anything, right? Does that make sense? And also because this has a free 3' end, you know already about DNA replication, this can now serve as the site for DNA replication, right? And so we have essentially a little bit of a recapitulation of DNA synthesis going on here, right? That's actually another important part. So the first part is breaking the chromosome. Second part is chewing it back, basically. So we have single-stranded DNA, and then that invades the other chromosome and serves as the basis for more DNA replication. And so as it does so, it kind of displaces this third strand, right, even further, right? Essentially, we've got like a little replication fork going on here, right, with just a leading strand being synthesized. And as it does so, this displaced third strand, you can see it's now starting to line up with this other strand down here, right? So there's now an overlap. And because these line up perfectly, right, base to base, and this displaced strand is now single-stranded DNA, right? It's now free to base pair with other complementary sequences. These two can now base pair together, right? And that's basically the process by which this recombination occurs, right? And so now we have another free 3' end. DNA synthesis is going to continue here, right, which is what you see down here, right? So now we've got it going in both directions, right? So we're using essentially both strands of this unbroken chromosome as templates for DNA synthesis, right? That essentially continues until that DNA polymerase hits the 5' end of the other, like basically the other broken bit of DNA, right? So this one is going to kind of keep displacing this intact strand until it hits the 5' end of this broken chromosome. And we're going to get the same over here. This is going to continue until it hits this 5' end. And when it does, you get the same process of backbone sealing using DNA ligase as you do in normal DNA replication. And now you have essentially this kind of hybrid, right, where you have a bit of original DNA, and then you have a mix of the 2 chromosomes, and then original DNA, and the opposite on the other side, right? And these are now joined together. These are called holiday junctions, right? And these have to be cut. Otherwise, these 2 chromosomes can never come apart, right? And this is done by what are called holiday junction resolving enzymes, right? So don't worry too much about where this is kind of done, right? It can be either done this way or that way. And it essentially just determines the site of crossover, right? Which can matter if you have a lot of different alleles that you care about. But if you look at this in the case of just 2 different loci, which are outside of this recombination zone, it doesn't make any difference, right? And so this is actually what it looks like if you look at a, what would that be? Probably a transmission electron micrograph, right? Using, what would it be? Gold labeled DNA would be my guess. Anyway, what ends up happening is you have part of the original chromosome, part of the original other chromosome here, and then a stretch of hybrid chromosome where you have one strand from each for a fairly short stretch. And that's how you end up swapping bits of chromosome. It's also how you end up doing it perfectly base by base swap, right? So you don't gain or lose not even one single, ni uno, base pair or nucleotide here, not even one, right? It is a perfect match. The only thing that happens is now we have a hybrid chromosome of both strands of the original one. Well, let's say this was be the unbroken one. Both strands of the broken one and 50-50 of each in this kind of recombination zone. And that's basically how this whole process works. Did I tell you about extremely radiation tolerant bacterium? I'm pretty sure I told the other class. I'm not sure if I told you lot. It's got a nickname, Conan the Bacterium, which I think is kind of funny. Probably none of you have ever watched Conan the Barbarian because you're too young. But if anybody has, please let me know. I won't feel quite so old. Yeah. Anyway, yeah, it's a super cheesy film, right? Like as cheesy as you can get. It's got Arnold Schwarzenegger in it, which kind of gives you an idea. But essentially, this bacterium, its real name is Deinococcus radiodurans, which basically means it's a bunch of balls which are resistant to radiation. And it can, I can't remember the exact numbers, to be honest. I'm not real good at that. Let's say we can be exposed to, let's say, 5 sieverts of ionizing radiation, like from a radioactive accident, let's say, before we start dying, right? Because it doesn't take an awful lot. This bacterium can actually live at levels over 1,000 times higher than that, right? So they could, if there's sufficient food, they could basically live in a nuclear reactor, right? Which is just astonishing. And one of the ways in which they do that is that they have, like 4 to 8 copies of their genome at any one time. And so basically their DNA is being broken constantly, and they are constantly undergoing homologous recombination to fix those breaks. Isn't that crazy? Anyway, so these are the kind of bacteria that I'm not sure about these ones, but there are others that actually live on uranium. They use that for their electron acceptor and cellular respiration, which is kind of crazy. Anyway, so that's pretty much that. I don't have anything more to bang on about. So the floor is essentially now open. So you can ask me any questions about anything that we've covered since the last exam, and I'll do my best to answer them. It is all now for you. Any questions that you may have. Hopefully I'll get at least one. So anything that you struggled to understand, that didn't quite make sense, anything that you want to check up on, any of that for the exam on Friday. Okay. Yeah, load me up, gentlemen. So different polymerases, not all of them, no. But the ones that illustrate important functions or differences, yes. So for procs, need to know what, oops. Don't worry, I don't have huge hands. It's just because my camera's down there. It kind of freaks people out sometimes. So you need to know what they do, like the main ones, right? For eukaryotes, need to know the difference between the one that starts, right, which has primase activity, and the ones that finish the job. Epsilon. Also, difference between primer removal and prox and ukes. Right, so essentially things that I've stressed, right? So the approach with DNA replication, and we'll be doing this with other parts such as control, gene expression, control of gene expression, things like that. Typically, I always cover the prokaryotes first because they're a little more straightforward. There are typically, not always, fewer moving parts, pardon me, and players involved. And so it's a little easier to grasp the basic principles first before looking at kind of a more complex or slightly more subtle version in eukaryotes, right? And so these different, sorry, I got itchy nose. These different polymerases really illustrate some of the key differences between prokaryotes and eukaryotes, right? And so if I've talked about those differences, which, I did, well, maybe not last class, maybe the one before that, I can't remember. Then you can be fairly confident that some part of that will be on the exam. Can't guarantee that it will be this. It might be like some difference in another function or something like that. But if I'm interested in you understanding those differences and using those differences to illustrate the process, then yes, you do need to know them. That help? Oh, hell no. Whew. No, we're done. We're done with that for now. So that pretty much ended with kind of non-Mendelian inherit. Oh, actually, ooh, hang on. Let me just double check the schedule. Oh no, there will be. Ooh, So essentially, oh, good job I did check. Good question. I'm going to roll that answer back. Honestly, sometimes I don't know whether I'm coming or going. So we still have some of the Mendelian and non-Mendelian inheritance. Well, actually, not the Mendelian. We have the non-Mendelian inheritance in this exam, right? So there's a bit of overlaps. I get confused sometimes. I think we finished all that by the first one. So yes, there will be some of that. It's going to be more in the context of different types of non-Mendelian inheritance, right? So maybe it's kind of like hard to answer that question specifically, but there will be an absolute ton of stuff on linkage, right? So you need to know how to do, and this isn't really probability, this is more kind of using numbers to work out distances. You need to know how to do a 3-point cross, right? So, stuff like that, and that's just a 2-point cross. You need to be able to do that really comfortably because you'll get at least one, maybe more of those to work through in the exam. And they take time, I know that, right? So it takes at least 5 minutes to work through one of those questions, right? And we have, 40 questions in an hour and a quarter. right? Which is, that's about 2 minutes per question, give or take. Now, a lot of the questions will be relatively straightforward recall questions, which you should just know, should be able to just pull them out of your head, right? Again, you don't have a lot of time to go looking up these questions on the internet, right? That's the downside of having an open book exam, right? There's the assumption that, well, if you can look this up, I'm not really testing your knowledge, right? I'm testing your ability to use Google. And so that's part of the reason for this being time limited, right? Why you don't have all day to do this exam. Because essentially what I'm rewarding by doing that is people that have studied hard and remember those things, right? So when you see a question, it's just like, oh, that one, that one, that one, right? They should be real easy. And that will then free up the time to spend more time on things like working out 3-point cross, right? And so that's really where the benefits of studying just like these DNA Pol 3, it's the one that does prokaryotic DNA extension. DNA Pol 1 is what removes primers and synthesizes DNA in the gaps. Easy peasy, right? So those questions will take you as long as it takes to read the question. That's how long it takes to answer it, right? 15 seconds, maybe 30 seconds, right? So that gains you a lot of time to spend working through the harder questions and thinking about the harder questions and doing a good job of answering those as well, right? And so if you don't have that basic kind of knowledge, then you'll be spending some of that time answering those questions and you won't have as much time to answer the hard ones, right? And so you might not do as well in those as you maybe could have. Yeah, so practice, make sure you know this stuff ideally off the top of your head, right? Don't rely on the fact that it's open book because that's not going to help you as much as you think it will. Okay, what else, peeps? Good question, Sophia. Yes. So you should be able to look at the genotypes of the offspring, and based on which ones are more common, you'll be able to figure out the genotypes of the parents. And based on those genotypes, then you'll be able to figure out whether those 2 or 3 genes are in cis or in trans, right? And so, for example, if you get parents which always have the highest number, right? Because if 2 genes are linked, you'll have an excess of parental chromosomes. If you have one which is one parent is recessive for one trait, dominant for another one, and you have another parent which is dominant for the first trait and recessive for the second, that tells you straight away those genes are in what? Cis or trans? If you have a parental chromosome which is either dominant recessive or recessive dominant, because they're going to be a mirror image. Is that going to be in cis or in trans? Trans, excellent, right. And so if you have parents which are both either dominant or recessive, that tells you that the original chromosome, the genes were in cis, right? And so you should, even without me giving you that information, right, which sometimes I will, maybe sometimes I won't, right? You should be able to just look at that and go, oh, the recombinants of this, the parents of this, the parents of this, which means it's in cis or in trans, right? Just from that data alone. And that also reinforces the kind of the 3 steps to 3-point cross success, right? Figure out which are the parents first, because that will tell you a lot. That will tell you what you need to know about the genotype of the parents, which is really important for figuring out, all the other steps afterwards. All right, next question. Jelena, you said you had a list. Surely there wasn't just one thing on it. Or if there is, that's awesome, right? Kind of works both ways. Okay, what do you think you need to know? I can tell you whether you need to know them or not. Here you go. Everything. Well, you have 2 days, Jimena. It's not a lot of time, admittedly. And don't forget, every question that any one of you asks benefits everybody else as well. So yeah, I mean, I'm always kind of careful generally in stating definitively the things you need to study most, because if it's in the course, then it's important enough to be in the course, right? I'm not going to tell you stuff which is unimportant, other than like some of the random tangents I go on. You don't need to know who Arnold Schwarzenegger is or what films he starred in, because that's just not important. Um, Especially for genetics. In general, it's not important, right? But generally speaking, I tried to make the exam representative of what we studied in class, right? So I'll make sure that there's nothing that we covered in class which isn't covered in some way in the exam, right? Not obviously that could be covered in different ways in the exam. Could be short questions, could be long questions, could be easy ones, could be difficult ones, right? But generally speaking, I try and make the proportions match, right? So if I spend half of that time talking about linkage, you're pretty much guaranteed we're going to have an awful lot of questions on linkage, right? Maybe not exactly 20, because there's only so many ways you can ask questions about linkage, but there'll be a bunch of them, right? Similarly, if we spend a bunch of time talking about DNA replication, all the different stuff on it, then you can expect probably more kind of recall questions, maybe, but also questions like what does DNA polymerase epsilon synthesize, for example, right? But also focusing on the principles, right? The concepts at hand. And so really, I mean, it's all important, to be honest. Otherwise, I wouldn't be teaching it. And for Jimena, it's difficult, right? Because I know that you have other things going on, and this is kind of right midterms sort of time, I guess. I can't really tell you how to apportion your time between one thing and another, but do study as hard as you can. One thing to remember is that you're not going to be doing this forever, right? And so even if you have to kind of sacrifice some spare time and maybe even time with family or friends or just kind of downtime, you know, come May, hopefully you'll go, whew, I got what I needed in genetics. Glad I don't have to do that again, right? Or yay, I got what I wanted in genetics. I absolutely love that course. You know, either way, it's great, but yeah, sometimes you just have to sacrifice a little bit on the way to get what you want. It's just hard work, essentially. Oh boy, that's tricky, gentlemen. I'm sorry to hear that. Can't you do both? Like study genetics, like at the wedding? Yeah, like look up from your textbook as the vows are um, spoken. That's a real shame. I'm sorry to hear that. Um, that's kind of tough. I've unfortunately, because I've been in the US, missed kind of all of the funerals for various family members that have died since we came here. Because it's very, very hard to kind of get back there, particularly if it's in the middle of a semester. So I can I understand that it's not quite the same, but hopefully you'll be able to at least live stream some of it, right? So you can kind of like actually see the process. That would be good. All right, any other questions? Questions? Okay, so don't forget, oh, okay, question. So unidirectional and bidirectional only applies to bacteria. And so I don't actually know why one would choose one over the other, but essentially it's like drawing around a circle in one direction with one pencil or both directions with two pencils. That's really like if you're going to be tracing around, I don't know, the bottom of a cup, let's say, right? Unidirectional is just starting at one point, kind of going all the way around until you get to that point. Bidirectional is starting at one point with two things and going around until you meet most likely opposite the starting point. But that only applies to prokaryotes. Eukaryotes are always bidirectional, right? And that's because you start at multiple points along a linear chromosome and you kind of start with a bubble and it gets bigger and it kind of until one of those replication forks kind of basically hits another one and those bubbles merge. The whole semi-conservative part is simply that you use one strand as template and one strand is synthesized. So each new molecule of DNA will consist of, pardon me, one old and one new, basically. Did that help? That was Sophia, I believe. Oh, crab sticks. Sorry, get chat back up. There we go, that's better. Yeah. Jelly, yeah, I mean, you need to know how it works in both. Well, you need to know how things work in both prokaryotes and eukaryotes, right? I don't tend to do a bunch of compare and contrast type questions because I just find them less interesting personally. So try not to kind of fall down that hole. But certainly the differences are useful one way in which you can study the processes themselves, right? So if you can remember how primer removal works in bacteria, right? That's one way of going, oh, hang on, it's very different in eukaryotes. Eukaryotes do it with da 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 da, right? And so similarly, you can compare primer removal with in eukaryotes with say mismatch repair, right? Because in some ways it's similar, right? Not entirely, but in some ways it is, right? So essentially, part of the reason for comparing the two different types of organisms and like looking at the differences between them is that that's just one way of learning how things work in the two different things, right? It's not that the differences are necessarily the most interesting part, right? Because, I mean, I guess maybe they are, right? But really, it's to use that as a way to remember and to learn how things work in the different systems, right? And that's no different from comparing how this works here and how it works over here, right? You know, what are these things which are shared? You know, so in both mismatch repair and in eukaryotic prime removal, a whole bunch of the RNA and DNA, in the case of primers, or just DNA in the case of mismatch repair, is removed, right? So that leaves single-stranded DNA, which DNA polymerase comes along, delta most likely, comes along and fills in, right? Which is also how mismatch repair is carried out as well, right? Obviously, there's a lot of differences between those two, but just being able to compare those two parts of the processes is one way of kind of connecting those things together in your head. right? And so that's another way to kind of think about studying, right? It's not just about like stuffing like things through your eyeballs until it's kind of like smooshed at your back of your head, and then you kind of try and pull it out in the same way in an exam, right? You want to put it in like one way and then put it in a different way and then connect that with something else and kind of make a network of ideas and information, because that makes it a lot easier to get that stuff back out again, right? Because you might not remember it in exactly the way I taught it, but you might remember it in a different way, which you put it in, which makes a little more sense. And that makes it a lot easier to get it back out again like that, right? And so, studying isn't a monolithic kind of activity. There's lots and lots of different ways in which you can get stuff in. And the more different ways you do, the better, by and large. Got, oh, I did have the chat up. Cool. Good. Any other questions? Don't forget, if you do have questions about particular parts of the material or whatever, just send me an email. I'm more than happy to answer any tricky bits the best I can. All right, everybody. So I think that's kind of ran us pretty much to the end of the class. Again, any questions you have, don't be shy. Just send me an email. I'll reply typically within a day is my aim. And I'll be around on Friday in the Zoom. I don't know, drinking a cup of coffee probably and checking my email. If during the kind of first half of the class when the exam will be open. So it'll be available from 9 a.m. on Friday until midnight the same day. You can take it at any time. But if you want to catch me for whatever reason, could be just last minute clarification or something, or you have a problem with the exam or whatever, I'll be in this Zoom for at least half an hour on Friday from 9 o'clock. Cool. All right. I'll see some of you later, and I'll see the rest of you maybe Friday, if not next week.